All right, everybody, I'm going to review some things that you're going to need to know for this test. First of all, you're going to need to know how to go from DNA to RNA, from RNA to amino acid using this chart or any other codon chart. You're going to also need to know that in protein synthesis, transcription, what it is and where does it happen. You're going to need to know translation in step 2. You need to know what it is and where does it happen. Awesome. You're going to need to know that all living things have the same 4 bases, A, T, G, and C. The only difference is the order of the bases. The order of the bases is going to give us different proteins. So once you switch the order of the bases, you're going to switch the order of amino acids, which is why a monkey and an ant looks different because the DNA bases are in different orders. Therefore, the amino acid sequence of monkeys and ants or bacteria is also in a different order. You're going to need to know that DNA replication is semi-conservative, meaning that half of the strand is old and half of the strand is new. You're also going to need to know that DNA is held together by weak hydrogen bonds so that they can be separated easily. Again, you're going to need to know that DNA is held together by weak hydrogen bonds so that they can be separated easily. You're going to need to know the bases that's in DNA and RNA. 3 big questions that you're going to see. Why do we need to eat protein? Well, our body makes 10 of the 20 amino acids that we need. So we have to eat proteins so that we can get the extra amino acids that we need. You're also going to need to know why DNA is needed in every cell, but we know that DNA makes proteins and protein is needed for everything. Why is it important for these bonds to be weak in DNA? We have to be able to break up the hydrogen bonds, the DNA molecule. When it comes to mutations, you must know that mutations happen randomly and in all of our cells. However, if a mutation happens to our eggs or to our cells, then we're going to pass that mutation to our kids. If you get the DNA that's mutated, then the entire protein synthesis process will be mutated, and then the trait is going to be mutated. So DNA is what starts. DNA is what mutated. DNA gets mutated, transcription gets mutated, translation gets mutated, then the protein gets mutated. And whenever the protein is supposed to mate, it's going to be mutated as well. You also need to know that on the instructions are very, very long. There's a initiator or a start codon that lets you know when to start reading the instructions. And there are stop codons. And these stop codons let you know to stop reading the instructions. You need to know the different type of genetic mutations. Substitution usually don't have a big effect on your body because one base is switched out for another. But anytime insertion or deletion occurs, the mutation is going to affect your body. You will need to know how to recognize tRNA. tRNA is always in the shape of its name, T. tRNA has 2 things that's important: anticodons that matches up with the codons and amino acids. You will need to know how to go from the anticodon to the mRNA for the DNA. And you will need to know the steps in order to make a trait. You will need to be able to use tRNA in order to get the amino acid sequence. You will need to know the difference between a gene, a chromosome, and DNA. You will need to be able to recognize which organic compound this is. If you do a process of elimination, you will get this right. Or if you just think about which unit we're in right now. All right, that's it. See you guys tomorrow, ready for your test. Don't forget to do your homework.